All right, we are now recording. So like I said, we're going to be starting with vision, and we're going to be looking more in detail at the eyeball as a whole, and then we'll look at more detail at the retina, where the actual rods and cones that do the signal transduction live, and then we'll look specifically at what's going on in the rods and cones to accomplish the conversion of light energy into electrical signals. Whew. All right, so eyeball. If you look at an eyeball from head on, we can, beautiful green eyes. There's supposed to be the iris. This is going to be the pupil. Eyeball. So this is eye front view. If we, cut it open so you can see the inside of it, it's going to look like this. This is a view that is going to look familiar from our lab. So this window, the clear window that kind of lets you let lights enter the eye, is the cornea. This white, I'm drawing it in black, but it's white. This is the sclera. Both the cornea and the sclera are what we call the fibrous tunic. So they're tough connective tissue. Right? The eyeball is actually really pretty tough. The sclera is basically like tendon. It's pretty tricky to pop the eyeball. Cornea obviously is a little different than the sclera because it has to be clear or else the image can't actually get into the eye. When your eyeball is actually in your head, there's actually a mucous membrane that actually kind of goes across the front of the eye. It has little blood vessels and stuff. Like when you look at somebody's eye and you see like those little blood vessels, those are actually not in the eyeball itself. They're actually in this mucous membrane that covers the front of the eye. It's called the conjunctiva. Conjunctiva. So it basically kind of seals, the front of your eyeball. Like if here's your eyeball and here is your eyelashes, your eyelids, I should say. Here's your eye. The conjunctiva kind of seals under your eyelids over the front of your eye. So this way, if you get some dirt in your eye, it actually is not going to get behind your eyeball. It's actually kind of trapped by this mucous membrane that kind of seals the front of your eye. When the conjunctiva gets inflamed, that's obviously conjunctivitis, and your eyes get all red and itchy and stuff. But yeah, that red, when somebody has red eyes, it's not the eyeball. It's like the blood vessels within the conjunctiva, this mucous membrane, which covers the eye, the front of the eye. Mucous membrane. All right, so the eyeball itself, though, we have the fibrous tunic, which includes the cornea, which is the clear window, the sclera, which is just that tough tendon-like rest of the eyeball. Then we're going to get the choroid, which is what we call the vascular tunic here. So let me erase my little eyeball here. So now we're gonna, and I'm gonna make a fatter line. So lining inside this sclera, there's this black layer called the choroid, and then it expands to what's called the ciliary bodies near the front. So now let me make my pen a little. So the choroid, one of the reasons it's black is just to reduce internal reflections. Like if you have light coming into the eye and it just bounces off all the walls, the image is going to get all kind of blurry. If you look inside, if anybody used to actually use film cameras, when you open up the camera and put the film in, it's always just flat black in there because you don't want light entering the camera to start bouncing around and messing up the image. So this choroid is just this kind of black layer in there. The ciliary body here does a few different things. The ciliary body is going to have different things. You don't have to know the specific names. There's the ciliary muscles, which are going to help with the focusing, ciliary processes, which are going to make the aqueous humor, which we're going to talk about in a few moments. So let's first talk about the ciliary body as a way to adjust focus. And actually, the choroid then extends to become the iris. I should put that in there as well. Again, because this is like a cross section, the iris is actually like a frisbee with a hole cut out of the middle. But if you look at a frisbee with a hole cut out of the middle, just like this, it just looks like two lines with a little gap. And always remembering that your pupil, the black dot in the middle of your eye, is actually not anything at all. It's just a hole. It's like when you look at the pupil and it looks black, it's just because it's really dark inside the cave inside your eye. If you take a flash picture of somebody and light gets in, it illuminates, and there's blood vessels in here, then you got red eye. So the pupil is not actually a thing at all. It's just a hole into a dark room. So ciliary bodies have these muscles which are going to adjust your lens. So again, we've seen this in our lab already. There are these little, it's called suspensory ligaments, which attach to the lens. And it's a little counterintuitive. Basically, what happens is the lens, if the lens is totally relaxed, if the lens itself has no stress on it, the lens tends to be more roundish, kind of more curvature. When you yank on the lens, it flattens out. You can kind of imagine that. If you have this thing with no pulling on it, it tends to just tend to being more round, but if you pull on the ends, it flattens out. So when your eye is relaxed, there's actually tension on the lens to make it flatter. That was what we talked about, like kind of distant vision in the emmetropic eye. The muscles that are in the ciliary body, these little ciliary muscles, actually, when the muscles pull, they actually release the tension on the lens, so then the lens will become more and more round. So when these muscles, there's little ciliary muscles here, muscles. When the ciliary muscles pull, they release the tension on the lens and the lens will get more round. So ciliary muscles contract. So that was accommodation. When you're trying to focus on something close, you are contracting these little ciliary muscles, which releases the tension on the lens. The lens ends up actually getting more round, and then it bends the rays more, and you're able to focus on things that are closer. The iris is like the iris diaphragm on your camera, the f-stop, to allow in more or less light. And there's basically going to be smooth muscle, which is the involuntary muscles. There's going to be smooth muscle that's more longitudinal. Maybe I should do maybe blue. So if you have the fibers that are going this way and they contract, it's going to dilate the pupil. You have circular fibers that are going this way. So here I have this blue, this is supposed to be smooth muscle. Smooth muscle fibers. And maybe actually this might be easier if I just, there are circular fibers. They're going to constrict the pupil. And there's going to be longitudinal fibers, which are going to dilate the pupil.
And those are all the ciliary muscles?
No, so this is not. This is smooth muscle in the iris itself. The ciliary muscles are the ones in the ciliary bodies that adjust the lens. So when people talk about the intrinsic muscles of the eye, there's two. Intrinsic muscles of the eye include the ciliary muscles, which adjust focus, and these muscles in the iris that adjust the diameter of your pupil. So that's two different muscles that your autonomic nervous system can control. It can adjust the ciliary muscles to adjust the curvature of the lens. It can adjust the smooth muscle in the iris to adjust the diameter of your pupil.
Okay.
Which division of the autonomic nervous system is going to trigger the circular fibers?
The parasympathetic?
Yeah, exactly. It's going to be the parasympathetic. So what kind of receptors are you going to find on the smooth muscle fibers of these circular fibers?
ACH?
They're going to be the muscarinic cholinergic receptors. Again, so like when you're going for an eye exam and they put those drops in your eye so they can like look in your eye, they're basically, those little drops are cholinergic antagonists. So they bind and block the cholinergic, the acetylcholine receptors on these circular fibers. So when the optometrist or ophthalmologist, whoever he is or she is, shines a light in your eye, these circular fibers don't get activated and the pupil stays dilated so they can look inside there. So the other thing we should talk about inside here, let me actually, let me add the retina in here just for completeness. And then I'm going to talk a little bit about the fluids that fill up the eyeball. So I'm going to give myself a little more room here. Okay, so we had basically the fibrous tunic, which was just the kind of tough connective tissue outer coating, the choroid and the ciliary body and the iris. It's all part, collective, we often call that the vascular tunic or the uvea. And then there's going to be the retina, which is like the sensory tunic or neural tunic. So the retina is going to be kind of lying right inside the choroid here. And again, we're going to look at the retina in more detail in a few moments. But before we get to the retina, the lens has to be clear, can't have blood vessels, obviously. The cornea has to be clear, can't have blood vessels, obviously, because otherwise you're going to be looking through spider webs of blood vessels. But there are cells that need nutrition and such that make up the lens and the cornea. So the lens itself has this layer of epithelium that makes these, they're called crystalline proteins that fold in this very regular way to make the lens. But there's actually kind of the same way we had CSF flow in the brain, where you had the choroid plexuses making CSF, which then circulated around and then drained into the arachnoid villi, into the sinuses. You've got the same kind of idea going on inside the eyeball. So the back of the eyeball, behind the lens, this is just filled with this more jelly-like stuff called the vitreous humor. So all in here. It's, again, it's kind of like this gumdrop. Again, if you've been in anatomy and you've dissected like cow eyes, it's like a big gooey gumdrop. This is called the vitreous humor. Or maybe I should. And this is jelly-like. However, the front of the eyeball is filled with this watery stuff, which they call aqueous humor. So I'm going to give myself some more space here, erase some stuff. So the aqueous humor is going to be in this space kind of in front of the lens. It's actually created by the ciliary bodies. It's kind of made by the ciliary bodies. And it flows around in here. And then it drains. There's a little canal. Sometimes they call it the canal of Schlemm, where the fluid then drains away. So it's being produced by the ciliary bodies, flowing around, providing nutrients and stuff, and then it drains at this canal, which is right at the junction of the iris and the cornea. You know, again, I'm drawing it like two little circles here, but it's actually like a donut, right? Because it's right at this boundary around the outside of the iris here. There's actually let me show you. I have a cool little video. It's not a video, it's a PowerPoint thing that actually shows this, actually kind of nice. So I'm going to put that on here just so you can see it. So like right here, this is kind of showing, oh, let me get my little pointer. Kind of back here, this is where you have that vitreous humor. This is kind of the back of the eye here. And this would kind of go all the way back towards the retina. This is the front of the eye here. Here is the ciliary body. So this is where we're actually making the fluid, making the aqueous humor, which is draining around, goes here through the pupil. Here's the space behind the cornea. And here is that little, they're calling it the scleral venous sinus. That's another name for it. But it's kind of more fun to call it the canal of Schlemm. And I think this might even, does it do it? Yeah. So it's like, here's the eye. Oh, look, we're making aqueous humor here at the ciliary body. It's flowing around. Oh, look, it's draining into the canal of Schlemm. So let me go back. What is the condition that's going to happen if you don't have adequate drainage? You're going to actually stack. You're going to actually, discard. You're actually going to start building up pressure inside your eyeball. So what is that condition if you actually start building up pressure in the eyeball because you have inadequate flow of the aqueous humor?
Glaucoma.
Exactly, that's glaucoma. So I wonder if I can, let me get my little pen. Yeah, so it'd be glaucoma. So glaucoma is when you have inadequate flow or drainage, so you get buildup of pressure. That pressure against the back of the eye, the retina, can actually start causing problems where the retina is going to start not being able to get the nutrients it needs and all. Another common eye problem is when the lens itself here starts becoming more kind of cloudy. Rather than being crystalline and clear, it starts getting milky. What's the condition if your lens starts getting kind of cloudy?
Cataracts.
That's cataracts, exactly. And if you have cataracts, you can um, have surgery where they will plop in a new lens. What's going to be the problem of just plopping in a new lens to replace the cloudy lens? Like, what does this lens do that's really nice for you that if you just put in a little glass lens that would not work so well? Anybody?
It wouldn't be able to adjust as easily. I
Yeah,
mean, it's just a glass.
yeah, exactly. It would not be able to adjust its focus. So one of the things that's, people have to decide is what, since they're only going to have a fixed focus, they actually need to decide. Sometimes people get lenses that are, um, for far vision, and then they just need to use glasses if they're going to look at things that are near. Sometimes people get lenses that are for near vision and then use glasses to look at things that are far. Or a lot of people, I know some people who have actually did it where they put a near focus lens in one eye and a far focus lens in the other eye. And over time, they've just kind of gotten used to their automatically just um, take in the image from one eye or the other eye, depending on whether they want to notice things that are near or far. So, so that's one of the things that happens when people have cataract surgeries. They have to make decisions about how they actually want their vision to be in the focus, their focus to be in the future. Um, because the lens, or actually because the cornea also doesn't have any blood vessels, the cornea is actually a really easy thing to transplant. Like if you will your body to science, one of the first things they harvest off of you when you come in with as a dead body is your cornea, because the cornea is something that's really easy to transplant on somebody else since you don't have to connect up any blood vessels or anything. So corneas are actually things that get transplanted a lot as well. All right. So everything we've been talking about is kind of like the big picture of the eyeball, but we haven't really gotten to the nitty-gritty of the actual sensory transduction in the retina. So what we're going to do now is do that. I'm going to get a different PowerPoint here. Let's get this one. So this is actually, this image in front of you is kind of an electron micrograph of the rods and cones. You can kind of see why they get the name rods and cones. Some of them look like rods. Some of them look like cones. They're not actually a pretty color like this. This is obviously just artificial coloring, but those are actual electron micrographs of rods and cones. Again, they're going to be sitting right along the back side of the eye. Let me get my little pointer again. Again, the retina is all along kind of the back. You know, we've already talked about the blind spot. There's this one area where all of the axons that leave the retina have to burrow out the back to form the optic nerve. That's going to be that optic disc or the blind spot. You know, we can even talk about what is light. Again, light is just electromagnetic radiation. There's basically these perpendicular electric fields and magnetic fields undulating through space-time, and depending on how fast they're oscillating, if they're in this really narrow range, if the wavelength, basically wavelength, let me get a little pen again. Wavelength is just what is the distance from peak to peak as these electromagnetic fields are undulating. If that distance here is somewhere between 400 nanometers, remember a nanometer is 10 to the minus ninth meters. So if it's somewhere between 400 nanometers and 700 nanometers, that's what we perceive as visible light. And again, we perceive it as visible light because we have receptors that will convert electromagnetic radiation in this range into electrical signals that go to our visual cortex. The same way we perceive the presence of different molecules as butterscotch or lilacs, we perceive the presence of electromagnetic radiation in this range of wavelengths as visible light. If you have electromagnetic waves that are much faster, you get into ultraviolet and X-rays and gamma rays and cosmic rays. They're the same stuff, they're just faster. If they're slower, you get into the infrared, the microwaves, radio waves. You know, radio waves are related to visible light the way that is related to it's just like lower frequency, higher frequency. But it's the same stuff of the universe. So our rods and cones are going to absorb and even what the heck light is. If you've taken physics, you probably know, we have nice, models and tools to work with describing light and how it functions, but it's weird. And sometimes you think of it as a wave, sometimes you think of it as particles, photons, or wavicles, or whatever. What exactly? It's energy, and it's it's very cool to think about, but it's also, the more you think about it, the stranger it gets. We've already talked about the whole idea of the lens focusing in lab. We don't got to do that anymore. So this is the retina. The retina is ultimately derived from the brain. While your brain is developing, we talked about how the forebrain became the cerebrum as well as the diencephalon. That forebrain also becomes the retina. The retina kind of starts branching off of the diencephalon and kind of migrates up into the front of your face to become the retina. So there's actually, these are actually all specialized neurons, and they actually do quite a bit of pre-processing of the visual image before it actually goes out into your optic nerve. So it's more complicated than the other sensors where you basically convert the sense into a stimulus and send it to the brain. Here, there's actually going to be lots of other stuff going on, which we're not going to spend too much time talking about. What we are going to do is spend time looking at the cones and the rods here. Again, these are the cells that actually do the actual transduction. They then release a neurotransmitter. They actually release glutamate. If you remember, glutamate is kind of a classic excitatory excitatory neurotransmitter, glutamate. Again, I'm writing with a mouse here, so I'm gonna seem really, really awkward. So the rods and cones release glutamate, which then talk to the next layer of cells, the bipolar cells. The bipolar cells talk to the ganglion cells. The ganglion cells are the ones that actually have axons that have action potentials. So these have action potentials. These are the things that are going to send the message to create your optic nerve. So
Sorry, you said that ganglion cells, what do they do?
they are the ones that actually are going to have action potentials that they send down their axons, and those axons are the things that exit the back of your eye to be your optic nerve. So if we go back to, basically the ganglion cells all send their, let me use a different color here. The ganglion cells send their axons, and all of those axons leave the back of the eye, and those become the optic nerve here, which is going to your brain. So ganglion, and one of the weird things is the rods and cones are actually more towards the back, and the ganglion cells are more towards the front. So the light actually has to go through the ganglion cells, go through the bipolar cells to get to the rods and cones. But because they are transparent, it really doesn't matter so much. Plus, when we get to that fovea, that center of the eye, the rods or the different cells, the bipolar cells, the ganglion cells are kind of folded to the side to kind of minimize the thickness of distortion. But the reason why you have the blind spot is that all of these axons are more towards the front of the eye, and then they have to burrow towards the back, which means there has to be an area where there's no rods or cones. So other animals, if you were like a octopus, octopus actually has the rods and cones towards the front and then the ganglion cells towards the back. And so then the light comes in, activates the rods and cones, sends the message to the back. You don't have a blind spot because the axons are already on the back. On a human, though, because the ganglion cells send the axon to the front and then it has to loop towards the back, you create this place that can't have any rods and cones. Again, I have no idea why it ended up like that. And as we saw in our lab, in general, you don't notice it because your brain just makes up stuff to fill in the space where you don't have the rods and cones in the blind spot. So back to here. I am not going to go into the details in here, basically. Some of the stuff that's going on here is doing what we call lateral inhibition. You can basically increase the contrast of your image. Like if you're having some area of the retina getting very active and it suppresses the circuitry on the side, it's going to actually increase the contrast. It'll make, if this is bright and this is not as bright, and the bright stuff is suppressing the stuff on the side, it means the less bright stuff is going to seem even lesser bright. So it's going to make boundaries look even clearer. So again, I think for right now, let's, I'll leave it at that. There is all sorts of kind of, contrast enhancement and stuff that's happening here with all this circuitry. This is going to be important. The fact that the rods and cones release glutamate. One of the things that is non-intuitive is they release more glutamate in the dark. When it's bright, they actually release less. So you might think like, wait, shouldn't they do more of releasing this neurotransmitter in the light? It's just the way it is. And because you have this more complex circuitry as you go further on, the retina knows. It knows if there's a lot of glutamate coming, that it's dark. It knows if the glutamate drops off, that we've got bright. So when we start talking about the details of the rods and cones, we're going to see in the dark, they release glutamate. And when we have light, they release less glutamate. And I should also mention, the only cells in the retina that actually have action potentials are going to be the ganglion cells. All the other things here are just going to be graded potentials. Glutamate's going to bind receptors, which are going to increase voltages. And as we increase or decrease voltages, you have more or less neurotransmitter release. But it isn't until you get to the ganglion cells that you actually have actual action potentials, which then go and become the optic nerve. So what we're going to do is we're going to look at rods and cones first kind of in a big picture and then in the very specifics about how does light and dark affect the release of glutamate. So let me get out of this more awkward thing here, and I'm going to go back to my whiteboard. So here's the eyeball again with the cornea. The rods tend to be more along the periphery of the retina, as we talked about last time. Periphery of retina. Obviously, light is coming in on the front here. The cones tend to be more in the center of the retina. Again, the very center is called the macula, and actually the center of the macula is called the Fovea Centralis. Fovea Centralis is pretty much pure cones, and your area of most acute vision. Part of the wiring here, part of the reason that the macula with the cones has the most acute vision is basically each cone basically ends up talking through a bipolar cell to a single ganglion cell. So each cone kind of becomes an individual message going down the optic nerve. So you've got that high resolution. If I activate this cone and not this cone, and then I activate this cone, my brain knows on, off, on. So it's got a lot of power to resolve fine detail. The problem is you need a lot of photons to like excite a single cone enough that it can activate this on its own. So again, these are only going to be active in brighter light. Rods, you have a whole lot of convergence. You can have like 100 rods that all meet up. That all meet up together to become, actually I should have it going. So there's a lot of convergence. What that means is even if you have very low level light, it all kind of adds up and you can still notice that there's low level light. But because you don't know which rod is actually talking, you don't get the detail. You don't actually know what is the fine detail. So remember at the end of class last time, we talked about how rods function in dim light, but they have low acuity. And this is part of the reason that's true. It's also why it takes them longer to respond. They kind of have to integrate across a number of rods to finally send the signal and say that, yeah, there's light. It's very low, but there's light. Whereas cones are getting this big dose of bright light and going zing, there's bright light here. Also, you can notice that the rods are on the edges versus the cones being in the center. In fact, when you're looking at stars up in the sky, I don't know if anybody likes to go stargazing. There's like a special trick. If you want to see a very dim star in the sky, you don't look straight at it. Why do you not look straight at a star, a dim star, I should say, if you want to see it in the night sky? So you're here and then looking up at the sky, the stars. Why do you not want to look straight at the star if it's very dim?
Because the rods are on the edges.
Yeah, because if you look straight at something, the image lands on this Fovea Centralis, which is just cones, which only responds to bright light, which means you're not going to see anything. So you're not going to see something really dim if you stare straight at it. The trick is you look to the side of the star, and then the image lands on the rods, and then all of a sudden you see it. It's really trippy. You'll probably, next time you're on a starry night, look off to the side, and you'll see like dimmer stars out of the side of your eye, and then look straight at the dim star, and you'll realize, holy crap, it just disappeared. And like look away from it, and you'll see it again. But if you put your gaze directly on it, it'll land on the cones, which don't respond to the dim light, and then it's going to disappear from your visual experience. So rods, I'm going to go back to another image here. I'm going to share screen. Actually, first I got to bring an image up. Give me 2 seconds here. Remote instruction, physiology. Oh, wait, that's not it. Wait, where did I put them? Dag nabbit. I just downloaded and stored a few images, but now I have to remember where I just stuck them. Did I put them in here? No. Oh, here they are. All right. So I'm going to go share my screen here. And what? No, that's not what I meant to do. Hold on. I just lost my control. So this is definitely not what you want to be seeing. Dagnabbit. Control Alt Shift. There it is. Share. This is what I want to share. There you go. So this is showing rods versus cones in terms of what frequencies. I talked about how visible light is light in this 400 to 700 nanometer wavelength. You have three different types of, three different kinds of cones. And I can, let me get my annotate, draw. So I have cones that can respond to these lower wavelengths, higher frequencies. These would be my blue cones. I have these ones that respond in this kind of mid-range, my green cones. These ones that respond in the lower frequencies, my red cones. So these are all different kinds of cones. And then this one here, this is the rods. So notice there's only one kind of rod. That's why they're monochrome. That's why you don't detect different colors. You know that something is blue using your cones because the blue cones are active and the green and the red cones are not. So your brain knows, oh, it must be blue because it activated the blue cones and not the green and the red. But with rods, there's only one version. So you just know, yes, there's light, or no, there's no light, but you don't have any information about what frequency that light is. From this picture here, you can get a sense, like if you've ever worked in a dark room or you work in other situations where you have like safe lights, where you want to have illumination, but you don't want to destroy your dark adapted vision. What is the color that the rods really don't respond to well at all? Anybody from look at, say it again?
Red.
Yeah, red, exactly. So like the safe lights are usually like, or if you have like a headlamp that has a low, like, a lot of these headlamps now let you go into this red color if you want to look at things when it's really dark, but you don't want to destroy your dark adapted vision. Normally, when things are really bright, when the cones are really happy, the rods are bleached out and all of the little molecules that detect the light are kind of overwhelmed, and it takes them like 5 or 10 minutes before they actually are fully functional again. But if you have dark adapted vision and then you use red light to make things a little brighter, it's not going to destroy your rod vision because the rods aren't paying attention to the red.
I have a question.
Yeah?
Last night when I was doing the little experiment, I noticed the red was really hard to, the red couldn't come into focus. It was hard to see the red. Is that the same thing? Like I could see the blues and greens in my room. I could adapt. The rods would adapt to those blues and greens. But when I was trying to focus on bringing the reds in my room into focus, that was
Right.
that light
So
color.
the rods don't notice red at all.
Yeah.
So
Okay.
so blue and green won't look blue or green. They'll just look kind of like illuminate. You won't see the color of them, but you'll see they're there. Whereas red actually doesn't even show up at all,
Yeah.
really.
Okay.
Yeah. So no, that's exactly what you would expect. So what we're going to do now to kind of finish up our discussion of vision is look at what exactly is going on in the rods or the cones in order to convert light into an electrical signal. And it's going to be very similar to what we've been talking about with receptors triggering second messenger pathways, except, instead of a receptor binding a ligand, there's going to be a conformational change of a molecule due to absorbing light energy. But it's going to come back to second messengers, cyclic GMP and phosphodiesterase, the enzyme that breaks down the second messengers, and all that fun stuff that we've hopefully feels kind of familiar now. So and then after we do that, we'll spend a few moments looking at the big picture of how the eyes are actually wired into the brain and into the visual cortex. All right. Let's go back to the whiteboard. Yes. Okay. So this would be like a rod cell. Cones are pretty similar. Everything we talk about rods is the same for cones. The only difference is the little molecule that acts like as the antenna to absorb the energy will absorb energy in different wavelengths. So there's this outer segment here. And in this outer segment, we are going to have these specialized little membrane proteins, which are going to be, so this is membrane protein, which we're going to talk about in more detail in a few moments. Opsin plus retinol. Again, we're going to talk about those in more detail in a moment. There's all these little folds just to increase surface area. So you have lots of these little membrane proteins here in this outer segment. Here on the axons here, these are the things that release glutamate. So these are going to be releasing glutamate down to the bipolar cells, the next levels of the retina. And like I said, when it's light, they're actually releasing glutamate, and when it's dark, they stop releasing glutamate. So it's a little opposite of what you might think. Other things that we need to know in this cell. Actually, let me give myself a little back. Come back, Mr. Cell. In the dark, there are sodium channels that are open. So in dark, sodium channels are open. So if you have sodium channels open, what is that going to do to the voltage on the membrane?
It's going to make it more positive.
Exactly. It's going to make it more positive. So you get what's called the dark current. And the membrane voltage, remember, a resting neuron is usually somewhere down around -70. we're going to have the membrane voltage is going to be around minus 40 millivolts. And if you remember, minus 40 millivolts is above threshold for a lot of these voltage-gated channels. So what's going to happen is this is going to cause calcium channels to open, which cause the exocytosis of glutamate. So in the dark, sodium channels are open. We have this dark current, which is, again, the sodium coming in. It's this positive current coming in. It makes the voltage of the cell membrane go above minus 55, which causes the release of glutamate. And that's how the retina knows that it's dark. It knows it's dark if there's this constant release of glutamate. These sodium channels that are here, these stay open due to the presence of the second messenger cyclic GMP. So if you remember, cyclic GMP is just one of the classic second messengers. What enzyme have we met that would break down cyclic GMP?
Is it kinase?
No, kinase is a thing that phosphorylates things.
Oh, well.
It was, go think about Viagra.
Oh,
Remember what did Viagra do? How does Viagra help a guy maintain an erection?
is it PDE?
Exactly. Phosphodiesterase, PDE, is the enzyme that breaks down this stuff. You know, Viagra was a phosphodiesterase inhibitor, so it kind of kept the second messenger up, which kept the whole signal going. So in this process, basically what's going to happen is light, the presence of light here detected by the opsin in the retinal up here is going to ultimately turn on a second messenger pathway that is going to ultimately turn on phosphodiesterase. And I'll talk about how that happens in a second. Light ultimately activates PDE, which is then going to reduce the amount of cyclic GMP, which is going to turn off the sodium current. And the voltage is going to go down. And then this thing's going to stop releasing glutamate. So that is going to be, again, it's kind of convoluted. But basically, this opsin in the retinal, when they see light, that is going to, we're going to talk about exactly what happens. When opsin and retinal see light, they're going to activate a second messenger pathway, which turns on phosphodiesterase. Phosphodiesterase is going to break down cyclic GMP. Without cyclic GMP, you're going to turn off the sodium current. Without the sodium current, the voltage of the membrane is going to go down. When the voltage goes down, the cell stops releasing glutamate, and that's how the retina knows that light has arrived. So I'm going to go get out of this crazy, messy drawing, and we're going to go back to the opsin and the retinal and look at them on their own. And so let's do that. So this is going to be the cell membrane of rod or cone. Opsin is this membrane protein, and I'm going to draw it kind of like here. You have this retinol, which is associated with the opsin. So opsin is a protein. Retinol is actually derived from vitamin A. So retinol is, and retinol is the thing that's ultimately going to be at the core of this whole thing. Retinol, when it's kind of in its kind of quiet form, is called cis retinal. Cis because it's got the, cis conformation is when the two parts of the molecule, the two sides are kind of folded up in the same direction. If it absorbs light, it changes its shape. Because say this is a conformational change, it's going to become transretinal. Exactly what frequency of light it absorbs to make this happen depends on the type of opsin. So different types of cones or rods have slightly different versions of opsins. So this whole system acts kind of like as an antenna that detects different frequencies. But the basic idea for rods and cones is always the same. There's this opsin and retinol structure here in the membrane. If it absorbs the appropriate light frequency energy, or light energy at the appropriate frequency, the retinol will flip into this trans conformation. And then what happens is, so light comes in, the retinol actually detaches, and now we have what we call like an activated opsin, which is going to trigger a second messenger pathway. Via a G protein. Wait, that's not it. Called transduction. So here in the cell membrane, there's also going to be this G protein as well. So, again, my drawing here is kind of messy, but this is very similar to what we talked about when we had a ligand. Remember when you had a ligand bind a receptor, and then that activated the G protein, and the G protein turned on the second messenger pathway by making cyclic AMP or something? This is similar, except instead of having a ligand binding a receptor to start the whole system, you have light coming in, absorbed by the retinol. The retinol flips into this trans conformation, and as it detaches, it activates the opsin. The opsin activates the G protein, which starts the second messenger system. And ultimately, that second messenger pathway, I'm just going to say, just turns on phosphodiesterase. And that is going to, normally I would have two boards, and this would not be so hideous here. But we could say, like, basically one, light enters. Actually, I'm gonna type. And that way it's gonna be way less ugly. Two, basically absorbed by retinal, by Sis Retinal, which changes to transretinal. 3 is detaches from opsin, hey you, opsin, and activates opsin. For activated opsin activates G protein, which is called transducin. It gets named transducin because this is, I think, before they even knew G proteins. It was one of the first things they found. It was actually really kind of wild the way they figured this all out, because all these molecules are changing and doing all this stuff. They made it really, really, really cold, and everything goes really slow when it's really cold. So they actually slowed everything down that they could actually keep track of what the heck was happening in here. So shoot, not transducin. 5, this activates second messenger pathway that ultimately turns on phosphodiesterase. Then 6, PDE is going to break down the cyclic GMP that has been holding sodium channels open. So then sodium channels close and membrane voltage goes down. This causes rod to stop releasing glutamate and is interpreted by a retina as the presence of light. So for on your exams, I can say a common question that I tend to give. What am I doing here? Oh, I didn't even know I could do that. A common thing that I do is, talk about, how are different senses transduced. I talked about how sensory transduction is such an important part of all of this. But I'm going to use a fatter, sensory transduction. Remember, that's kind of what all this is about. How do we take some stimulus, whatever it is, whether it's light, whether it's the presence of a chemical, how do we turn it into some electrical signal? That's that transduction, translating into an electrical signal. So, for taste buds, how is the presence of sugar converted into an electrical signal for the nervous system? Anybody?
Taste buds, like getting into the receptors on taste buds.
Yeah, in taste buds, you have a receptor on that taste cell, that gustatory cell. It binds the sugar. When it binds the sugar, it ultimately opens up little sodium channels, which increase the voltage, which then ultimately sends an electrical signal to the brain, right? touch receptors. How do touch receptors actually work? How do they do sensory transduction? It's going to be similar. If we go back to the very first week of class, remember what are different things that can gate channels, that can make channels open or close?
Mechanical.
Yeah, exactly. Mechanically gated channels. So in a touch receptor, on the little sensory endings, you're going to have little mechanically gated channels, little channels that open when the cell is getting stretched. And as those channels open and let sodium in, it's going to create a positive voltage, which then is going to ultimately create a signal to the nervous system. So for the sense of touch, you've got mechanically gated channels that are opening to create a voltage change across the membrane, which becomes the sensory transduction. For taste or smell, you have ligand gated channels that are binding, like say sugar or something, to open up channels, create a voltage change, which is then the transduction. For vision, it's a little more complicated. The light is going to change cis to transretinal, which turns on this whole second messenger pathway. But ultimately, it's going to be about opening and closing channels, which change the voltage on the membrane. And that's, again, the sensory transduction. So for when we get to the hearing, which is the next one we'll be looking at, we will see there you have also mechanically gated channels in the hair cells. These things, as the little structures in the ear vibrate, bend in response to the air vibration, it's going to open and close channels, which create voltage changes, which are ultimately going to be interpreted as hearing. So does that make sense? I do want you to really think about what is actually physically happening that converts the stimulus, whether it's bending of your skin or sugar on your tongue or light coming into your eyeball, and what is the process where it actually becomes a voltage change? Because the voltage change is the only thing that your brain is going to be able to understand. So does that make sense? Yeah? Are there any questions about all this? I know this is a lot here. No questions at all? All right, so how about if we take a break and let's start our lab. Let's see, it's 9:34. How about 10 o'clock? We come back into this. I'm going to just put this on hold at 10 o'clock. We are going to start lab, and we're going to start the rods and cones lab. Yeah.